All right, so now we are at the point where we're going to talk about more organs of the endoderm, and this primer is going to focus on lung development. Now, for the objectives in the outline, I need you to understand the formation of the definitive endoderm as we've discussed before. Now, remember, prior to this primer, we've talked about pre-implantation development and implantation development, gastrulation, and the formation of the germ layers. And again, one of those germ layers is the endoderm. That's one of the first germ layers to form, okay? And the organs that come from this, I do want you to learn. If you've already looked at the other endoderm primer, I told you I would like you to learn the organs that come from the endoderm. Now, again, understand the formation of the definitive endoderm. Identify the regions of the endoderm gut tube. What are those regions? You know, the foregut, midgut, hindgut. Identify which organs arise from which region of the endoderm gut tube. So, you know, obviously you don't want to say on an exam or learn that the lungs form from the hindgut. That's not what happens. Understand the development of the lungs from the definitive gut endoderm. That's the focus of this primer. Lung buds and the respiratory diverticulum, primary and secondary bronchial buds, terminal bronchioles, respiratory bronchioles, terminal sac, primitive alveoli. These are things we're going to go over. And we're going to briefly explain here some of the basic molecular mechanisms that initiate and regulate endoderm organ development. One of the reasons why we're going to talk about some molecular mechanisms is that the mechanisms that form the lung are very similar for many of the organs that come from the endoderm. All right, so that's why we're going to touch upon it here. We will discuss in more detail the molecular mechanisms of lung development in lecture. Okay, the first animation I have here is a very short animation, and what it's showing you, it's giving you an orientation of where the lungs begin to form from. It's basically way back at the base of your throat, okay, in other words, and and you can see from this animation where the lung begins. And this is the pharyngeal arch region, which we'll talk about much later. We'll get into a lot more detail in head and neck, the head and neck module. But it is important to know that the pharyngeal arch region, the base of the throat, is where the lungs will develop from. Okay, so we have an image here. As you can see the image here, you've seen this image before, and I'll use the cursor here to point things out. Human development of endoderm is basically a tube within a tube body plan. A point I really want to make clear. You are all pretty much tubes within a tube, all right? And the yellow portion of the endoderm is what we're highlighting here. Now look at the respiratory diverticulum. It's highlighted in blue here, an A. It should of course be yellow, but they've changed the color just to orient you with what we're pointing at here. All right, and if we go higher magnification in B, all right, we see that respiratory diverticulum. It's a bud of endoderm that's coming off of the pharyngeal arch region, just way at the back of your throat, okay? And the pharyngeal pouches are here, the pharyngeal arch region is here, and you can see the buccopharyngeal membrane right here, opening of the mouth eventually. So we're going to focus on the respiratory diverticulum here. All right. So as you can see in D in this image, in the initial formation of lung buds and laryngeal development, weeks 4 through 12, D is that same image that was in the animation, right? All right. So look at the laryngeal orifice. All right. That's into the computer. screen or into the as you're looking at this. And if we go back to A here, that is where we're looking at. So the respiratory diverticulum is here, all right? And basically it protrudes from the foregut, and the first thing that happens, it sets up a proximal, where the cursor is here, distal axis, all right? Very important axis. Consists of two distinct epithelial cell populations. So there's a proximal bronchiolar epithelium and a distal respiratory epithelium, okay? I know it says respiratory epithelium, but no, it is not capable of oxygen exchange at this point yet, obviously, but it will become. Those cells eventually will bud and bud and bud and become alveoli. The proximal distal axis is very important in signaling, okay? So those cells at the proximal distal axis, where they are, will secrete different signals in order to enable the lungs to grow. In a few days, the tip of the respiratory diverticulum has enlarged, followed by division, you can see in B, into a right and left lung bud in B and C. Okay, so there's been a bifurcation of that lung bud into a right and left lung bud. Now, in D, concomitantly with that lung bud formation, we have the development of the primordial pharynx, all right, where the trachea is going to attach and you can take breaths in. The esophageal ridges grow. All right, those ridges are growing. All right, and esophageal tracheal ridges fuse, resulting in two structures, and that's going to be the esophagotracheal septum and the laryngeotracheal orifice. All right, that's that orifice right there. Okay, now again, what this is showing you in D, what the Roman numerals are pointing to are the pharyngeal arches: 1, 2, 3, 4, and 6. 5 fuses with 4, and there is no pharyngeal arch 5. Okay. If we go into this in a little bit more detail, all right, what you're looking at, where my cursor is, right there, you see that? All right, all of that tissue is endoderm. All right, all of the tissue that's inside of those pharyngeal arches, all right, that is going to be mesoderm, all right, and neural crest. Now, remember what neural crest, where that came from? Came from the development of the neural tube, those cells off of ectoderm. So neural crest cells are from the ectoderm, okay? But a lot of the tissue in here is going to be mesoderm. All right, now if we take a look at this in a little bit more detail with the arachnoids swellings come from mesoderm proliferating under that endoderm. So the blue that you see here is endoderm, but in the mesoderm underneath of it in arches 4 and 6, okay, that's proliferating and it's communicating to the endoderm, the arachnoid swellings. Okay, so as the swellings proliferate between arches 4 and 6, they'll proliferate around the laryngeotracheal orifice. By week 6 into 7 or so, that orifice is transformed into a T-shape opening, and that's bordering the epiglottis. Okay, so we have see in B, a T-shaped structure, and we have a primordial epiglottis that will become the epiglottis. All right, so the underlying mesoderm and neural crest cells are rapidly growing, and they are also communicating with the overlying endoderm to also tell it to rapidly grow. And we'll talk about that in lecture about those signals that are causing that. All right, so the endoderm will proliferate. All right, endoderm proliferation of the primordial epiglottis closes the laryngeotracheal orifice at about week 7.5, and by about week 10, the orifice is recanalized and is now a primordial glottis. Okay, so At about this stage of development right here in B, we have a primordial glottis. All right, that's all we need to talk about for now with respect to the development of that region. Okay, now I just want to briefly go over the innervation. All right, so we have cranial nerves, 10 of them. All right, and I'm not going to get into a lot of detail with respect to the cranial nerves. I just want you to know that they exist and they are important for regions of the head and neck. It's the innervation of the larynx, all right, the pharynx that becomes the larynx. 10th cranial nerve innervates the derivatives of the fourth arch, okay, so the fourth arch, and the superior laryngeal branch of the 10th cranial nerve, and the recurrent laryngeal branch of the 10th cranial nerve innervates the sixth arch. Okay, so we have the superior laryngeal branch of the 10th cranial nerve with arch 4, and arch 6 is the recurrent laryngeal branch. So the 10th cranial nerve is the important cranial nerve here that is innervating the region of the larynx that allows you to breathe, and the furthest back portion of your tongue to be innervated. Okay, so switching gears a little bit now, let's get into the development of the lung itself. So we've talked about the development of the pharynx and the larynx, all right, the back portion of your throat that allows the air to come in and out. Now, of course, we're going to talk about the lungs. So the buds are proliferating, the lung buds are proliferating into the mesenchyme, all right? Here, remember I said mesenchyme is a descriptor of a cell type. It just so happens that the mesenchyme here happens to be mesoderm. All right, I don't want you to mix up the two terminologies, but it is growing into the mesoderm. These buds are growing caudally and laterally into the pericardio-peritoneal canals. Okay, so the canals that are surrounding the heart. All right. The mesoderm covering the buds and bronchioles become the visceral pleura. That's what it's called. You can see here the visceral pleura. That's the mesoderm that surrounds the buds. These secondary buds represent the future lobes of the lung. 3 on the right, 2 on the left. All right. Once the secondary buds are well formed, they are known as secondary bronchioles. And those secondary bronchioles divide dichotomously, forming 10 tertiary bronchi on the right. All right, let's see if we can count these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, something like that. That's how they were drawn here. And 8 on the left. So from the table here, we have, in essence, 4 stages of development for the lung. There are some texts out there that have 5 stages, but we're going to focus on these 4 here. All right, so we have the pseudoglandular period from 5 to 6 weeks, cannulicular period 16 to 20 weeks of development, terminal sac period 26 weeks to birth, and then the alveolar period. Look at this, 8 months to well into childhood. Okay, and the definitions of those are on the right-hand side of the table. So let's get into this in a little bit more detail. So the purpose of the pseudoglandular stage, and that's week 5 to 16 or so, to grow and expand terminal bronchi into the surrounding mesoderm. All we're having is constant budding and bifurcation of those buds, and then bifurcation of those buds, and so forth and so on. The mesoderm overlying the endoderm of the lungs becomes a visceral pleura, as I've said before, while the parietal pleura overlies the body cavity. Okay, so those mesoderms, we'll talk about those mesoderms in the mesoderm lecture and mesoderm primers, but those are the mesoderms and how they associate with the endoderms of the lung. All right, we have the cannulicular stage. We have tertiary bronchioles are growing, dividing, differentiating into respiratory bronchioles. okay? And you can see that over here, these respiratory bronchioles here. And you can see they're beginning to associate with or coming in close contact with the capillaries that are approaching the lung. And of course, another purpose of the canalicular stage is to grow further within the surrounding mesoderm. Now, some respiratory bronchi can differentiate at this stage into terminal alveolar sacs capable of gas exchange. That's going to be closer to week 26 or so, not really much earlier than that. And by five and a half, 6 months, vascularization of the lungs is well underway. So those blood capillaries are in close contact with those respiratory bronchiole. And so, in some circumstances, by 5 and a half, 6 months or so, the lungs may be able to support the baby outside the womb. Okay, and we'll get into what happens at those premature stages if we have a premature birth in the lecture. What can happen detrimentally to breathing with respect to breathing air. All right, so we have the terminal sac stage, week 26 to birth. And what's the purpose? To firmly establish the blood-air barrier. That's what's going on here. So endothelial cells, all right, at terminal alveolar sacs change their cell adhesion proteins. All right, they change the proteins mostly into adhesion junctions, things like that. These changes allow that cuboidal shape of endothelial cells to flatten. So I'm over here with the cursor. I'm pointing to those thin squamous epithelial cells that came from those cuboidal cells. Now I'm pointing to right there. All right. It's just that they change. One of the changes that occurred was the adhesion, cell adhesion proteins that allowed this to happen. What that does is enables really close contact with the blood cells, okay? With the capillary, excuse me. So these new cell types are known as type 1 alveolar epithelial cells, and their flattened phenotype allow intimate association with capillaries. Right? It creates, in essence, creating that large surface area for gas exchange. All right, so now we go to the more specialization of the cells with respect to the terminal sac stage. All right, so 2 to 4 weeks before birth, it now becomes another population of cells, and that would be the type 2 alveolar epithelial cells. All right, these endothelial cells, they do remain cuboidal, or they can become pseudostratified, and the main purpose is to produce and secrete phospholipid-rich fluids known as surfactants. Okay, surfactants and soaps are not that different from each other, and we'll talk about that a little bit more in lecture as well. But the main purpose of surfactant is to coat the entire terminal alveolar sac and lower surface tension at the air-alveolar interface. Without surfactant, the baby will undergo severe breathing problems. All right. So, known genes involved in making surfactant. This is, I put this in here because it's important to know for a OBGYN, pediatrician, neonatologist. Was there enough gene expression in order to make surfactant? All right, enough surfactant. So we have thyroid transcription factor 1, hepatocyte nuclear factor 3. There are glucocorticoids, thyroxine, and of course surfactant A and B. That gene is obviously very important for making surfactant. We need to turn that gene on. Okay, so we get to the alveolar stage, 8 months to 10 years. All right, 10 years of age. All right, your lungs are developing. So obviously, to provide complete adaptation from aqueous, a dependent gas exchange in the placenta to a terrestrial environment. 3 adaptations must occur. You must have surfactant production. Without it, you can't breathe. proper differentiation of endothelial cells into epithelial cells capable of a gas exchange. All right, so you have to have complete differentiation. What happens if you don't? You can have certain types of diseases that occur. We can talk about that in lecture. We have to have establishment of pulmonary and systemic circulations, of course. We'll talk about that in the cardiopulmonary portion of the lecture and the primers. So, alveoli form up to age 10. 17 generations of bud division, so 2 to the 17th power occurs by month 6 in the fetal development, and you'll get your final shape of your lungs by about month 6 or so. An additional 6 or 7 bifurcations will occur, divisions will occur postnatally. All right, so we have quite a bit of division lung development happening postnatally. All right, let's talk a little bit about some pathology related to improper respiratory system development. So TEFs, tracheoesophageal fistulas. Anatomy of most of these fistulas will occur soon after birth, all right, when first feeding, for example, or when the baby's simply swallowing. Fluids spill over into the trachea and into the lungs. So take a look here, all right? So here's the blind ending part of the esophagus. Fluid spills over, it goes into the lungs, okay? And how would you fix that? We can just surgically fix that here, all right? But it would be a surgical procedure and very inclusive, or it's going to, you really have to go deep within that infant in order to fix that problem, okay? So TEFs are associated with a family of defects usually. So if you see a TEF, you're gonna have to look at some other things too. Vertebrae anomalies, anal atresias, cardiac defects, all right, the TEFs themselves, esophageal atresias, renal anomalies, and limb defects, and it's collectively called a bacterial set of defects, okay? So if you see a TEF, you're going to, as a pediatrician or an OBGYN, you're going to have to look for these other potential problems. All right, so we have the classic respiratory distress syndrome, or hyaline membrane disease, it's otherwise known as. It is a common cause of death in premature infants. At least 20% or so result from insufficient surfactant production. So the alveoli collapse upon exhalation, and the treatment would include surfactant replacement therapy. So you have a premature birth. One thing you're going to have on standby is a syringe full of surfactant. And in essence, you're going to need to, as that baby breathes out for the first time, if there's a problem with breathing back in because all the alveoli and the bronchioles have collapsed, you're going to have to force surfactant into the bronchioles, all right, so that you can open up the airways. Now, you can have a glucocorticoid treatment during pregnancies to accelerate lung development, all right, and to produce surfactants. That's one potential. If you know you're going to have a premature birth, you could do that. Oligohydramnios occurs from insufficient amniotic fluid in contact with the lungs. Remember, the embryo or the fetus must have the amniotic fluid bathing the lungs. Right? Areas with no amniotic fluid or little amniotic fluid will be underdeveloped. Congenital lung cysts is another problem, and they'll form as large bronchioles are dilated during development. It looks something like this over on the right, the picture that I put up here. All right, so on the right-hand side, you'll see a region of dilated lung cysts, okay? So they can be large, viewed by radiography, all right? Can give lungs a honeycombed appearance. Drain poorly and can result in chorionic infections after birth. All right, so those are just a handful of problems that you could see as a pediatrician or an OBGYN. These are all or a neonatologist, and I've given you kind of the most common anomalies that you could see. And when you go into your rounds, you'll see you could see many uncommon problems as well. All right, so in summary then, definitive endoderm arises from ectoderm. All right, more precisely, it arises from primitive ectoderm. Let's be clear about that. All right, and it's primitive endoderm that has ingressed through the primitive streak. Embryonic folding, three-dimensionally, gives rise to the tube within the tube body plan. The inner tube becomes the gut endoderm. The gut endoderm gives rise to not only the lungs, but the GI tract, the tongue, liver, pancreas, gallbladder, and parts of the pharyngeal arches. The lungs develop from a specified region of the foregut, starting as a diverticulum, becomes bilobed, and quickly develops a proximal distal axis. That proximal distal axis is very important for signaling and developing a proximal bronchiolar epithelium and a distal respiratory epithelium. The larynx forms concomitantly with the initial and later development of the lungs. The 10th cranial nerve innervates the derivatives of the larynx. Maturation of the lungs occurs beginning as early as week 5 of development and proceeds through early adolescence. And we can have problems with lung development if, for example, there isn't enough amniotic fluid bathing those lungs. All right? The balance of that amniotic fluid with the lungs is important. Too much or too little fluid can cause major defects. Some of those defects are in a group called vectoral defects, all right, and they can cause atresias and fistulas. Remember, this primer is only a summary of lung development from the endoderm, all right? It is a little bit more detailed than other primers because, as of now, this is the only information you will get for lung development in the respiratory module. All right, you can go back and look at the lecture that we had, and it will be posted in Scientific Foundations 1, where there is more information about lung development. So emphasis is placed on the organs and fetal anatomy that I think medical students will see in gross anatomy. You could see some of this in gross anatomy, and that way you can go back and forth to understand where these structures came from and how they structured, why they structured, and when they were structured. All right. So good luck, and we'll see you in the next primer.